Good morning, good afternoon, and good evening to everyone joining us from around the globe. Welcome to Cyagen's webinar, Fulfilling Your Research Needs, Cyagen's over 16,000 knockout and conditional knockout sperm repository for expedited discovery. My name is Sydney, the International Product Marketing Manager here at Cyagen, and I will be your host for today. Here at Cyagen, we understand the credible pressure and critical demands faced by biomodel researchers today, from the need for speed in model generation to the absolute necessity for consistent and high-quality data, all while managing tight resources. This webinar is designed to directly address those challenges and provide you with innovative solutions. Today, we have an exceptional speaker who is deeply familiar with these intricacies. Please join me in welcoming John Merritt, Scientific Account Manager at Cyagen Biomodels. John holds a Master of Translational Medicine from UC Berkeley and UCSF, bringing a unique blend of academic rigor and practical experience to the table. With a strong background in preclinical assay development, John has contributed significantly to the biotech landscape, having worked on CAR-T clinical trial and oral biologic drug delivery before joining Cyagen. In his role here, he specializes in helping clients design sophisticated custom animal model strategies, particularly within the dynamic fields of cell and gene therapy. Today, John will be sharing invaluable insights into how Cyagen's extensive knockout and conditional knockout sperm repository, combined with our advanced technical solutions, can truly transform your research workflow, accelerating discovery, enhancing efficiency, and ensuring the highest quality of your animal models. Please feel free to type any questions you have into the chat box at any point during the presentation, and we will address them during our dedicated Q&A segment at the end. Without further ado, let's turn it into John Merritt. John, the floor is yours.
Nice to meet everyone. My name is John Merritt, Scientific Account Manager here at Cyagen. Sydney said before. We basically make and design custom animal models for customers, and I'm happy to begin my section of the talk here while Sydney figures out her internet connection. So let me go ahead and get started. And this webinar is titled "For Fulfillment Your Research Needs, Cyagen 16,000 KO cKO Sperm Repository for Expedited Discovery." And we call it expedited because essentially we've already made the mouse, and that you're just basically doing IVF and then getting the heterozygous mice to get started. We think it's a better way of doing research as opposed to the traditional way, which is performing a pronuclear injection every time. We'll kind of discuss a little bit in more detail why that is. But yeah, like I said, I'm John Merritt, and you can email me at johnmerritt@cyagen.us. And if you have any questions, you could type it in the chat. Sydney will get to you, and after this webinar, we'll do a quick Q&A session. Ask your questions there. But the first question that we get from customers is, why sperm banking? So in essence, we perform sperm banking because of economics, essentially. That traditionally generating a knockout mouse model requires a lot of time-intensive steps, from the single guide RNA design, vector construction, pronuclear injection, and then back crossing, just to generate an F1 generation line. And it's faster and cheaper to just not have to reinvent the wheel every time you want to make a new model. And for projects where you generate a mouse model, you maybe make a discovery, you publish, but then you don't necessarily want to have to upkeep that live colony for, years or even decades for people to reproduce. You can just cryopreserve the sperm, and then 10 years later, if somebody wants to reproduce your study or want to continue your work, it'll be readily available, ready to go in just a few months. So in essence, we sperm bank because we maintain a sperm bank because it's more economical than doing a live colony, and also it allows researchers to reproduce your work on a faster timeline than having to do a pronuclear injection. And also, um, from a quality control and reproducibility standpoint, starting from sperm bank is better than having to redo the work from single guide RNA. So there's actually even better reproducibility and quality control in that. So let's talk about how sperm banking is better, not in terms of just terms of speed and cost, but also it allows you to generate multiple lines simultaneously. So you can come to us and get multiple, let's say you want to make a multi-gene knockout of like 3 genes. Instead of having to do 3 pronuclear injections in series, we would, or even doing in parallel, we can do 3 of them IVF in parallel. And so that's just allows researchers to have a more flexible platform. And if they want knockouts of 3, if they want 3 knockouts, we'll get them 3 heterozygous mice done in parallel. So on our end, what happens is we first start off with um, scanning through literature and finding genes of interest we find genes that are being studied by scientists. And then in our facility, we go ahead and design guide RNAs and do the vector construction and essentially perform the microinjection, the pronuclear microinjection in our facility, and then generate the F1 heterozygous mice in our facility, and then we do the quality control. We don't operate like a repository like JAX, where they accept donations from professors. Everything we do um, is crafted by us, and so we want to control and maintain the quality in our facility. And after we perform the pronuclear microinjection, um, we generate founder pups, do the back cross, generate the F1 heterozygous males, and then collect sperm and cryopreserve it for future customers. And, while originally maybe, nobody's interested because we have like, 16,000 of them and it's impossible to like know exactly what people want before they want it. When someone does want it, we just pull out the sperm, do sperm recovery, and then afterward, and just generate mice immediately. And so we use in vitro fertilization to generate the line again, although we'll be talking a little bit more ICSI, which is another way we perform like IVF that is better and allows greater conservation of like rare biological resources. So this right here is a video of us doing the first step, which is performing the pronuclear injection. And you're going to see here, there's like any pronuclear injection, there's 2 nuclei here. And what not all of them like show that in this video footage, but here. And we can also do cytoplasmic injection, and we can also do blastocyte injection for ES targeting projects. But I'll just show you the video here. It's kind of cool to watch. And then we just do this hundreds of times until we get a founder. And for certain common mouse models like C57 Black 6, FEB, we guarantee the genotype. So we'll just keep doing pronuclear injections until we finally get like a founder line that works for you. These are for custom projects. Um, but in essence, we're very confident in our ability to perform pronuclear injections. And for any project that you want done where we perform this for you, if it's on a background strain that we're familiar with and we worked with before, we guarantee the genotype. And if we don't get the genotype, then we offer a full refund or we redo the pronuclear injection. So we don't charge per diem per injection like other companies do. We when we give you a strategy for a project, we guarantee the genotype, and if we don't think we can, then we just tell you that and just let you know that this is probably a riskier project. That's common for immunodeficient mouse models. So yeah, we perform prenuclear injections, and we also do blastocyte injections for ES targeting. And also, it's good to note too, is we also perform intraplasmic sperm injection. So um, this is in replacement of IVF. We can also do IVF, and that's perfectly acceptable for most of the time. However, for um, mice that have low fertility or there's genetic edits that affect fertility, sometimes we recommend ICSI. So that way we can conserve more of the mature egg. And so basically in this procedure, we just literally take a micropipette. We're like sucking up the little sperms, and then we're removing the tail to prevent damage. And then we I think I'm gonna get three of them here, and then we go over to the mature egg, and then we inject it into the cytoplasmic region of the egg. So that's getting the sperm, and then here it is, injecting into the egg. And so going through the egg wall into the cytoplasmic region, injecting one sperm, and then we aspirate a little bit of the cytoplasmic region out just to confirm that we indeed injected the sperm into the cytoplasm. And we just do that over and over again. And like I said, both IVF and ICSI work perfectly well. But we recommend ICSI for mouse models that have low fertility. Yeah, it's just fascinating to watch it. So here, move on to the next slide here. This is just a table of showing how ICSI is better at conserving rare mouse genetic resources. Um, 4 different projects. We compared IVF with ICSI and found that we had a higher fertilization rate with ICSI. So it's about 80 to 85% fertilization rate relative to like less than 10% with IVF. So in terms of like resources, it's cheaper to do IVF because essentially you're just like putting sperm in an egg in a petri dish. But we get better results, better outcomes with ICSI. So that's something to consider. So with our large sperm bank, 16,000, we have developed a pretty robust repository management system surrounding it. And the best way to figure out if we have sperm for something is, of course, check our website, and we keep that updated regarding sperm. However, if you'll notice on our website that sometimes it says we have live mice, and it's very difficult to track live mice just because it's a lot of mice in our facility. So the best way is just to ask us, and then we'll go and check the vivarium for you and get the date of birth if we have it. Um, I will say, we have a lot more sperm than we do have mice, so it's pretty likely that we only have the cryopreserved sperm. Sometimes we do have mice, and we like for a knockout mouse model, and then if we do, we'll let you know. And we sell it pretty reasonable price, like per mouse, and happy to get you a quote for any of our live mice. And the best way to check if we have live mice available for a knockout is to check our flash sale. Flash sale is essentially just leftover mice from a previous project or from a large contract research project. And whenever we get an order for like to make 100 mice, so to speak, we don't actually make 100. We usually make 120 or 130. The extra 20 or 30 is meant for backup. And then the 20 or 30, if we have the the right to distribute it. We'll just sell it on a per mouse basis. So we can get you quotes for that, and you can check Cyagen flash sale. If you Google it, you'll be able to find it. And this is just a photo of one of the kind of repositories that we have of our cryopreserved sperm. This is also, I think, a great photo because it kind of shows just how logistically difficult it is to keep sperm. A lot of folks ask us, if you have sperm, why don't you just ship me the sperm? And we can certainly do that. We can ship you the sperm of male F1 heterozygous mice. The issue is that we have to do it on liquid nitrogen, and that's actually more challenging to ship than live mice. Because live mice, you just ship it in a crate, but with cryopreserved sperm, it's a little bit logistically trickier. We could certainly do it, but sometimes we don't actually have enough cryopreserved sperm to just ship out sperm only. We have to replenish the sperm repository every time we pull it out. So basically, we have only a few vials of the sperm, and so we need to replenish our sperm stock every time. So sometimes we can ship you sperm directly, but if everyone did that, we wouldn't have any more sperm left. So sometimes we ask that we generate the N1 heterozygous males back again to replenish the line. And like I said, the best way to find out if we have sperm is to check cyagen.com/mouse atlas. It's we just updated our website. We're pretty happy with it. And you can easily go and type in whatever gene you'd like. And we probably have like a knockout or conditional knockout for that gene. It also just can link you with other disease models that we have that are more, better validated. We have like a human VGFA mouse model for wet age-related macular dystrophy degeneration. Sorry, and we have like other like DIO models or immunodeficient models. We have pretty much every model that we have. You can look up on our mouse atlas, and I'll kind of show you that as I go through our example today. So the I wanted to also talk about how we make a knockout mouse model. So people ask us when they send us an inquiry for a knockout mouse model, what type, how did you make it, essentially, right? And so let me go back. We first search through literature and see what do people want being made, right? What's the kind of the what's in the market right now? What are people interested in studying? What are the rare diseases that folks are, trying to make gains on? And so we first find genes that are in literature that are relevant to scientists, and then we just look up the gene on NCBI, MGI, Ensemble, like we try to figure out what's already known, what's already done, because we don't want to make a mouse model that already exists. And then we choose a model strand. Looks like we have a question here. Oh no, never mind. So, and then we choose a model strand. So the model strands that we pick are the model strains that we pick typically are C57, Black 6, 6J, or 6N. Those are the two main ones. And sometimes we'll do bulb C. Sometimes we'll do this in rats if people want to, if there's demand for that. Sprague-Dawley, Long Evans, most likely. Excuse me. And when we pick a gene to knock out, there's every gene can make multiple transcripts and multiple isoforms, right? So we typically work with the canonical sequence, or we try to reference some literature when we make a knockout, and then we pick the exon that we want to select for. So picking an exon to target in the knockout strategy is very important. If we could do the whole gene, we'll do it. But sometimes we cannot because we're constrained on how much we can knock out. We can only knock out 20 kilobase pairs at a time with the global knockout. And if we wanted to make a conditional knockout, we're even more constrained. We can only flox around 6 kilobase pairs. So yeah, we kind of have to be selective about which exon we pick. And I'll kind of go into more detail how we pick our exon. And then we make the guide RNA, we design guide RNAs and we do a sequence analysis, and then we determine whether if we're going to use CRISPR or we're going to use ES targeting. And usually we only use ES targeting if CRISPR is not viable as a technique. That is like there's no exons that we can pick that would work. Most of the time we can use CRISPR, it's not an issue. But there's occasionally some niche projects where it's like we have to do it the old school standard ES targeting method. Um, yeah. And then we check for lethality or fertility defects. So not all knockouts will lead to viable mice that can reproduce. And so we have to be careful of that. If we do find that a project will lead, knockout will lead to lethality, then um, we don't do that global knockout, right? We do the conditional knockout. We recommend that you do like a Cree breeding, tissue specific, inducible knockout, and we'll let you know. So that way, it's like we're not going to waste your time with mice that don't end up existing, right? So this next slide is on how we pick an exon. So this is kind of jumping off the first slide here. How do we pick an exon? And the second slide here is just the kind of criteria that we use to pick an exon. These aren't like hard and fast rules. These are just like how we think about picking out exons to knock out for our customers. And remember, we're doing all of this before the customer buys, right? So it's like we're trying to kind of get a guess at what they're looking at and what they would want. And so we need to have like a democratic sort of criterion or criteria to picking out exons that kind of can kind of satisfy everyone in the scientific community as a group, right? And so the first thing we look for is frame shift inducing exons, frame shift inducing mutations. So we're targeting exons that are non-triplicate, right? And introduce an early termination codon via frame shift or like a nonsense mediated, and that results in nonsense mediated decay. So we're trying to pick out exons that are non-triplicate that result in early termination codons. And then after that, we check to see if that exon that we picked is present in all transcripts possible, all of the isoforms, to prevent any sort of like other isoform coming in and functionally replacing and rescuing the knockout, right? And then when we pick an exon, we try to prioritize kind of the beginning, the 5' proximal region. So that ways we can ensure disruption um, as early as possible. But we don't try to do it so early that we're like targeting the start codon. We don't target the start codon region because we still want the translation to be initiated in the start codon, but then we want that start codon to go off and to translate nothing. And so that kind of gets into if we can't find an exon that is compatible, then we try to just target whatever domain is like most important according to literature. So that requires a literature search. But then people kind of debate over what is the most important domain. So we try to stay away from that. We try to just try to take a purely genetic approach to this knockout and like not try to figure out which domain or which protein region is most important that will likely lead to like whatever phenotype. It's kind of hard to make these strategies because everyone wants a different phenotype from the same exact knockout. And so we try to just make a purely genetic knockout in that sense. We also try to ensure that there's the flanking intronic regions uh, are still somewhat preserved so we can maintain some genomic stability. And kind of going into criteria 6, we're limited in what we can knock out. If we can knock out the whole thing, we knock out the whole thing, you know? But it's not always achievable given the CRISPR technology. We could certainly do it with ES targeting. So if we find that there is functional compensation and there's all these tricky kind of mechanisms that the mouse has to restore function, then we might just take an ES targeting strategy and just knock out everything, just end to end, when we don't want any sort of question about the any sort of fragment coming in and compensating or something. So sometimes we'll do that if we can't meet all these criteria. Um, Criteria 7, obviously criterion 7, we don't want to disrupt anything super important like critical regulatory elements. We want to try to maintain as much of the mouse as possible while still leading to like a knockout. And we consider a knockout as in like absent Western blot, so like no protein, right? And we still try to allow some translation to happen. Like we want the start. What we worry sometimes, and sometimes what happens is that if you try to knock out the start codon, that the mouse still has a way of translating and starting from a different start site. And so essentially, like we want the translation to be initiated, and then we want like a little bit of a chunk of amino acids to come out, and then we want like it to stop and then to decay. We don't want to just take out the initiation because then the mouse might just initiate somewhere else, and then we have the same problem. So yeah, also genes overlap with each other, so we try to exclude other genes that try to maintain some buffer zone between genes. Anyways, if you have any questions about how we pick an exon for a particular strategy, best to just like contact someone like me who goes off and, makes these strategies, and we can kind of talk about what you're looking for in particular. We try to just produce like genetic knockouts, and we don't necessarily have like a phenotype in mind or a particular research field in mind when we make these. And so sometimes it can be difficult to like make a one size fits all knockout for everyone. And a lot of the times in the past, we used to make these just by hand, right? We would go through all the criteria, look up on NCBI, do the literature research, and essentially we got very tired of doing this, as you can imagine, for all the genes in the mouse genome. So we essentially started scraping through literature data, like phenotype databases, other types of databases to essentially train a computer model to make strategies for us. And that's been very, very helpful for us. So some of these strategies are generated by AI, and we do still look at it at the end of the day. It's just that there's so many genes in the genome, we can't possibly do them all. And we have started generating strategies using our in silico model, and we've have gotten gene editing design pretty well optimized at this point. Um, so still we look at it at the end just to make sure, and we double check everything. But this has been enormously helpful to generate strategies, and it's allowed us to basically offer free strategy designs, and also has allowed us to do it uh, very fast. So if you want us to generate a strategy, even if it's a custom project that has nothing to do with our eBank, we still do this. We use we use AI to basically generate these strategies. We sit down, we talk to you, and you've paid no money up front. You've just gotten a free consultation session on how to generate. Sometimes what people do is they'll come to us, we'll generate the strategy for them, and then they make it themselves. And we essentially are doing free mouse consultations, and we're fine with that. It's like it's just we just enjoy making people strategies at the end of the day. So this is sort of how we end up doing it. And it's like I think the main takeaway is that this system automatically generates gene editing strategies in just 10 seconds. So it's just like um, I think gone are the days of like people working, designing strategies, like getting the right guide RNAs, all this stuff, making vectors and all this stuff. Like it's just now like generated through artificial intelligence, and we're much happier for it, and we can focus on other things. Uh, so let me kind of walk you through how this whole thing works for LAMP2. It's a gene of interest. So let's say you're interested in buying um, LAMP2 knockout, right? So what I have here on the top is the gene transcribes. There's some alternative splicing that sometimes happens that leads to three main transcripts: LAMP2A, LAMP2B, and LAMP2C. So let's say, you're studying this and like you're like, okay, I'm interested in making a knockout, and by default, we'll just knock out the LAMP2A, 2B, and 2C all together. And so here, what it looks like um, on here's the track, and you're gonna find that these three um, transcripts or these three isoforms are different in the last exon, which is the 9A, 9B, and 9C, right? The beginning is all the same. And so we've selected the canonical sequence, and we have now, and I can look it up for you. Let's see. So this is Mouse Atlas. You come in, type in Mouse Atlas, LAMP2, and let's say I'm looking for the flocks, right? So you just type in LAMP2 flocks. And so you'll see here, we got LAMP2 is located on the X chromosome. We designed, looks like a CRISPR strategy, as sgRNA and ssDNA were designed using nuclease technology. And so you just go through, see, okay, we got four strategies here, four flock strategies. And this one right here, go to the strand description. This one targets exon 9 of LAMP2, and it's meant for the LAMP2A isoform. Okay, well, I'm not looking for that. I'm looking for the full knockout, all the isoforms. So this one, okay, this one's targeting the LAMP2B. And then this one right here, let's check out the third one. Okay, this one's targeting exon 3. So that is likely the global knockout there. And just go over, download the document. You can go and get the strategy from us as well. And you can also check out the rare disease data center here. So this is the LAMP2 lysosome associated membrane protein 2. Looks like it's involved in, what disease is this involved in? Let's look it up. So here's the gene expression pattern, related mouse models, literature references. So this is well studied here. People are, looks like, studying alophatic vacuoles and cardiomyopathy in this mouse model. So LAMP2, this is adjacent genes next to it. Interesting. Okay. And there's also, let's see if I can look up rare disease data center. Oh, rare disease data center. Go through here, type in LAMP2, and you'll see that this gene is associated with lysosomal glycogen storage disease. This is a rare disease that this gene is associated with, and then you can just click LAMP2. Here's the there's 622 different mutations, 90 of them are pathogenic. So there's a lot of different types of mutations here. And then obviously if you knock it out, you're creating, let's see, Dinan's disease. So I've never heard of Dinan's disease, but it seems pretty rare. It looks like it's X-linked. That would make sense because it's located on the X chromosome. Let's see, categorization, medical symptoms, um, abnormality in musculoskeletal system. And so this is all like in mice. So as a patient-facing doctor or doctor in medical research, you'll be interested in studying this if you want a reproducible mouse model that uh, has has Dinan's disease. And yeah, so you can also kind of go through the reference. Here's the publications in it. Looks like the first one, or looks like people are publishing it a lot actually. And here's another publication here. Primary LAMP2 deficient causes X-linked vehicular cardiomyopathy and myopathy. And you can kind of look up the paper, read through it. This allows you to kind of just put a connection between rare disease and genetic knockouts, right? You could start off with the gene, see what the disease is, or you can go search a disease and see what the knockouts are for it. So, and this kind of allows you to, this is sort of what we put together the rare disease data center to try to, as a translational tool, like seeing which knockouts lead to which diseases and how to help people try to get the disease phenotype that they're looking for with the knockout that they're buying from us, essentially. So yeah, that's kind of a walkthrough on how to pick a knockout and how to go and purchase one if you'd like. And so, like I said, um, this disease is likely lethal, right? Because it's cardiomyopathy. So you'll probably want to get the Flox mouse model for this one. I don't think you would want to buy the global knockout. I think homozygous, it's embryonic lethal. So, unless you only plan on working with the heterozygous global knockouts, I recommend that you get the conditional knockout. And then we have Cree mice. If you buy Flox mouse, we'll throw in the Cree breeding pair for free if we have it as live, livestock. We keep most of our Crees live, but just ask us about what do we have live and whether it's Cree ERT2 or iCree or any of those other types of Crees, and we'll get it for you. So a lot of the times when customers reach out to us, they ask, what if my target or how long can you keep sperm viable for storage? This is an important question because sometimes what we do is we'll do a custom project for a client, but then we'll offer them a discount, and in exchange for that discount, we'll have distribution rights. So if you come to us and you're saying, I'm studying this disease and I think this knockout will lead to this disease, um, you could buy it and just own the model outright, and we'll just be basically offering you a fee for service, right? Or we can kind of enter, if it's a gene that we're interested in studying too at Cyagen, then we'll basically split the cost of the generation per nuclear injection and all that. But in exchange, we'll have the right to distribute and sell it. For that 50% discount, you essentially get someone who can cryopreserve sperm for you and keep it indefinitely. And you can just do your research and get half off essentially. But when we do this cryopreserved sperm storage, people ask like, how long can sperm remain viable in storage? And we check every 5 years, and we'll replenish the line usually by then. So we sample, and then we have a seed bank and a production bank. So we have ways of storing these long term, theoretically indefinitely. But we do perform QC checks on any of these just in case. And the second question we get is like, what if my target gene is not in, Mouse Atlas, or the particular floxing strategy is not there, or, well, we recommend that you go and talk to us, and then we'll let you know if we're in the process of making it. And if not, because there's so many genes out there, we may not be actively making it right now. We are happy to make it for you, and then we'll just split the cost, right? And then, you get the mouse model. You're one of the first people actually to get it, really, when you think about it. So that gives you the ability to publish before your predecessors, and then we get to just add it into our sperm bank. And then if anyone wants to reproduce it, you just tell them, I got it through Cyagen. They have the cryopreserved sperm. They come to us. We do the sperm recovery IVF, and then you have now a collaborator who can also reproduce your work without you having to maintain a live colony in footing the cost there. So yeah, great way to do research. And we're running a lot of promotions right now. So this is a good time to get this started, you know. We're running buy one, get one free. That's right. If you want one, we will get you two for the price of one. And if you yeah, so this ends September 30th. So feel free to reach out to me about it. I'll line you up with whatever. Our conditional knockouts are more expensive than our knockouts, and so the buy one get one free um, only applies for you'll get the free mouse of equal of or lesser value, right? So we kind of talk more about what that means, but essentially, if we're doing IVF for one conditional knockout and you want to throw in a second, we'll just put it in that shipment for free. So, want to appreciate you for your time. Thank you very much. Um, I understand it's pretty early for some of the folks in California, but I hope you have a nice weekend. Thank you very much for your time.
Thank you, John, for this insightful and comprehensive presentation. And it's clear that Cyagen's advancement truly offer a significant leap forward for researchers. Now we've reached the highly anticipated Q&A portion for our webinar. We've already collected some excellent questions that have come in during your talk, and we will now dive into those. John, are you ready for some questions?
I am.
Okay.
Yes.
So the first one would be, what are the main advantages of using ICSI over standard IVF when generating mouse models, and when should I choose it?
Well, both ICSI and IVF work perfectly fine. And so for most research needs, you want to go with IVF just simply because it's more affordable. But we do ICSI only when there's fertility issues and we want to conserve, mouse, uh, we want to conserve eggs essentially.
Thank you, John. And the last one comment is, if I'm designing a lab 2 knockout, how do you handle creating a complete knockout versus targeting specific isoforms, especially with alternative splicing to consider?
Yeah, with alternative splicing, design, we can just target the exon specific uh we can target the exon specific to a particular isoform to just get you like a knockout for only that isoform. Like we can just knock out just lamp uh 2B on exon 9 for you to get that specific knockout for you. We can also just knock out all three of the isoforms by targeting earlier exons in the 5' proximal region. It's like I think exon 3. But yeah, regardless, when we design these strategies, we take that in mind.
Thank you, and hope that answered your questions. And the third one is regarding the conditional knockout. The question is, I'm trying to optimize my flocks and pre-breeding for conditional knockouts. Do you have any tips for making that pipeline more efficient?
Yeah, we actually do. We've been rolling out what we call ES Cres. So these are essentially embryonic stem cells that have Cre, and so we can introduce the flux edit into those Cres and just work on it on the ES cells, and then perform a blastocyte injection, and then get you flux Cre mice right off the bat. So that's one way of making it faster. We only have certain like Cree ES cells, so like we don't have that many of them right now. But if you're interested in doing a Phlox Cree, it might be faster, honestly, to do it in ES cells. That might we might be able to skip a few generations.
That's really insightful. And the next one also regarding the conditional knockouts. So the question is, if my knockout is lethal or my conditional knockout study is delayed, what are my alternatives?
We recommend that you make a conditional knockout if it's lethal. And conditional knockout can be either inducible or just tissue specific. So you'll have to pick a good Cree for that, and we have Crees that we can provide you.
Thank you, John. And the last question is, CRISPR is everywhere now. So what makes Cyagen's approach truly different or better for my research?
We've just optimized this to a point where we can start offering it, buy one, get one free. So we just have a much more optimized platform than most of our competitors. And we're able to offer like custom projects for like 12K or like 13K. And we're also able to offer, IVF on our cryopreserved sperm for like 8K, and we can do buy one, get one free. So it's just like what makes us different is that, we're like an academic supplier and that we have scale.
Thank you, John, for those thoughtful answers and for sharing your expertise with us today. And a huge thank you to all our audience for your engaging questions and for dedicating your valuable time to join us. We hope this webinar has provided you with valuable strategies and demonstrated how Cyagen's knockout and conditional knockout sperm repository and comprehensive services can empower research, accelerate your timelines, and deliver reliable results. Remember, Cyagen is here to support your discovery journey. We encourage you to visit our Mouse Atlas platform at www.cyagen.com/mouseatlas to explore our vast collection of models. Don't forget our exclusive buy one mouse model, get one free offer for EU and US academic clients, detail of which can be also found at our official website's resource panel. Should you have further questions or require customized solutions, please do not hesitate to contact us directly. Our team is always ready to assist. Thank you once again for your participation, and we wish you productive research and look forward to partnering with you in the future. Have a wonderful rest of your day.
Thanks, Sydney. Take care.